This is AP Biology Enzyme Catalysis Lab. Today you're going to be working on a lab that involves the enzyme catalase and determining what kind of reactions it has under several different conditions. You'll be using several different kinds of materials for this lab, and I want to go ahead and point those out to you first. You'll be using the enzyme catalase. Catalase will be in the smaller cup, and this larger container will have ice in it. Catalase denatures at room temperature, so you have to keep the catalase at the ice temperature all the time. Do not take the catalase out and let it warm up. Another component that you'll be using in this lab is going to be hydrogen peroxide. Hydrogen peroxide will be in this cup. You'll also have a syringe that's labeled hydrogen peroxide. For this particular lab, it's very important that you read the labels 2 or 3 times before you actually use them to make sure that you're using the right syringe for the right component. Because if you don't, you ruin the lab and have to start over. Another component that you'll use is sulfuric acid. You'll have a container full of sulfuric acid here. Sulfuric acid is an acid. It will hurt you, so be careful. You also have a syringe that is labeled specifically for sulfuric acid. Use this one for only the sulfuric acid. The final component that you'll be using is potassium permanganate. You'll have a small container of potassium permanganate. Potassium permanganate reacts with the hydrogen peroxide and turns it pink in the presence of the hydrogen peroxide. The lab is divided into 4 parts. The first part, you will do some basic testing of hydrogen peroxide and catalase just to see what kinds of reactions take place. When hydrogen peroxide breaks down, it breaks down into water and oxygen. The oxygen is going to have bubbles, so one of the reactions that you're going to look for is whether there's bubbles or not. Catalase is found in nature in beef and potatoes and avocados. So we could be using liver, beef liver for this, or we could be using chicken liver or potatoes or avocados. I'm going to be kind to you and let you use avocados because beef liver is kind of gross. Anyway, so one of the first things you'll do is test the avocado with the catalase and see what happens. Another thing you'll do is boil the catalase in the test tube and then react it again with some hydrogen peroxide to see what happens. Be careful when you're handling the test tube, because if you handle the test tube in the hot water, it will burn you, so be extra careful. If we're looking at your lab papers, page 1 is instructions for activity 1. Then you have some procedures on page 2. Page 2 is kind of confusing about where those components go in your lab write-up. So let me just go through each one of them and tell you where they go. Basically, 1, 2, and 3 under procedures, they are procedures. Even though you're filling out blanks and you're filling out observations and that kind of thing, and that's normally going to go in data. In this case, because it's just procedures and you're just kind of basically seeing how the material works, we're just putting that in procedures. Then underneath that, you have the effect of boiling on enzyme activity. 1, 2, and 3, again, just count them as procedure. Put them all in procedure. And then on page 3, you have testing for catalase in living tissue. Again, just do it all in procedure. You have steps 1 and 2. Those are procedure, and put your answers for each one of those components in the procedure portion. The next thing we have on page 3 is your background to activities B, C, and D. Please make sure you read through that material thoroughly, because in order to do activities B, C, and D, you've got to know how to do this titration protocol. It's very important. So read through that. If you have questions, of course, make sure you ask me those questions before you get started. Activity B is determining baseline. Baseline is your standard. You've got to know what the baseline is before you start, because otherwise you're not going to know what the standard is, and therefore you won't know what kind of reactions are taking place. So do your baseline. If you do your baseline on one day, then you may have to redo the baseline the next day if you don't go on to the next activities. Activity C, you're going to set up on Wednesday, but you're actually going to do the actual work on Thursday. So go ahead at the beginning of the lab and set up activity C, because you will do that overnight. There is a cup labeled H2O2 overnight, because like I said earlier, hydrogen peroxide decomposes naturally into water and oxygen. It's just that the enzyme catalase makes it happen faster. So what we're going to do with the overnight one is just put some H2O2 in there and just see how much of it has decomposed overnight. Activity D is the one where you'll be doing most of the work, and this is probably the most complicated of all of them. So please make sure that you're paying attention to what's going on in this procedure. The first thing you're going to do is you're going to have these 5 cups. The cups are labeled with seconds on them. We have 10 seconds, 30 seconds, 60, 120, up to 180 seconds. You're going to put hydrogen peroxide in, then you're going to add catalase. You're going to wait 10 seconds and put in sulfuric acid. Now 10 seconds is not a whole lot of time, so please make sure that you have your H2SO4, sulfuric acid, ready to squirt into that 10-second cup as soon as the 10 seconds is over. Counting your time is very important in this lab, so make sure you have your timer out before you start activity D. Otherwise, you'll just have to start over, and I know you all hate to have to start over. So 10-second and 30-second, have that H2SO4 ready to go, because the 10 seconds and 30 seconds goes pretty fast. The 1-minute one, the 120-second and 180-second, you've got a little bit more time, obviously, before you have to deal with those. Once you've added the sulfuric acid, these can stay overnight because you've stopped the reaction. Nothing else is going to happen. The catalase is done. The hydrogen peroxide is taken care of. Nothing else is going to happen to these cups. So if you have to stop, you can stop at that point and continue this part on Thursday. If not, you're going to do titrations. You have a cup called titration. Titrations do, what they do is they tell you how much H2O2 has been consumed, how much is still in the container. So we're going to take a small portion of each one of these and put it, and it tells you in the lab how much of each. Put it in the titration cup. Then you're going to take potassium permanganate, and you're going to fill up this special syringe with the potassium permanganate. Make sure that you read very carefully how much you put into this tube. Then you're going to, again, with your titration cup, don't do this in your individual cups because you'll have to start over. So make sure you put it in here. You're going to have this syringe at a certain point, mark it down how much you're putting in there, and then very carefully, drop by drop. You don't want to put it in there too fast. Drop by drop, and after each drop, swirl. Drop and swirl, drop and swirl. It'll start to turn pink, but as you swirl it, the pink color is going to go away. Then you drop again, and it'll turn pink, and the pink color will go away. And then suddenly, it'll turn pink and stay there. It may even turn purple, but it's going to be there, and as soon as it does that, stop. Take your reading. That's how much H2O2 is left. Follow the directions. If you get all the way down to the bottom with your syringe and it still has not turned pink, go ahead and fill it back up with the potassium permanganate to a certain level. mark down how much you're using so that you have a total of, for instance, 5 milliliters, and then you start dropping again. This is tedious. Just be aware of that fact. Let me go back, page 4. Page 3, the background, that is not procedure. You do not have to include that in your procedures. Page 4, you have your materials, you have your procedure. The baseline table, that is data. Then you have number 7, that's procedure. Then on page 5, you have activity C, you have your procedures. Table 2 is data. On activity D, you have your procedures. It tells you how to titrate the time trials. Make sure you've read through that material, because after each transfer, you've got to wash your transfer syringe. Table 3 is data. Then you have analysis of results on page 7, 8, well, 7 and 8. 7 and 8, those are your analysis questions. And I think that for the most part that tells you what you need to do for the lab. Of course, you have to read the lab in order to fully get the entire procedures, but I've given you the basic idea. So that finishes AP Biology Enzyme Catalysis Lab.